Nandesh, we are going to start the human physiology also. So high weightage chapter, everything we are taken. Okay, fine. Don't worry. We are doing the human physiology also. Let us wait for one more to join. Then we'll start. Okay. Okay. So good morning, everyone. Today we are going to continue the cell cycle, and the biomolecules, right? Okay. So before going to start the class, can you tell me if I'm audible and screen is visible, guys? So then we'll start the class. Okay. Fast. Fast, fast. Very good morning, Melita. Okay, guys, fine. Thank you. Thank you. Let us. Yeah. So as I told you in the last class also, this chapter is very, very important chapter. And it is only this unit is carrying the 40 marks. Okay. So, and topic also easy to grasp, right? Okay, guys. So then here is a question for you. The question is, which one of the following structure is not found in the prokaryotic cell? In the last class, we have read the prokaryotic cell, right? So now you answer me fast. I'm going to give you only 10 seconds, guys. Okay. So I'm going to give you only 10 seconds. Answer me in the chat box. Answer me in the poll also. Yeah. So student from the YouTube, you can answer me in the chat box. Good morning, Lakan. Answer me in the chat box, Lakan. Very good, Nandish. Yes, Preetham, very good. Yes, correct, guys. Yes. So the correct answer is nuclear envelope, right? So nuclear envelope is not present in the prokaryotes, right? Because in nuclear envelope is a membrane bound, right? So no membrane bound organelles are present in the prokaryotes, right? So that is the reason nuclear envelope is the correct answer. We have studied about the mesosome in the last class. This is act as a mitochondria in the eukaryote, in the prokaryotes, right, guys? So it helps in the respiration and the cell wall synthesis. Then coming to the plasma membrane, obviously guys, plasma membrane is present in both eukaryotes and the prokaryote. And the only one organelle which is present in the both eukaryote and the prokaryote is that is ribosome. That is also we have studied in the last class, right? Okay. Then there is another question for you. Cellular organelle with the membranes are. Student always use the technique to solve the question. Okay. So use the elimination technique. Eliminate which are the not correct answer. By seeing only one organelle, you can eliminate the most of the option here. Try guys. Lakan, answer me in the chat box. Very good, Nandish. Yes. Very good. Yeah. So see here, guys. So cellular organelles with the membranes are. First of all, we will tell you whenever you talk about the whenever you talk about the ribosome and the centrosome. So these two are not having the membrane, right? So we will eliminate the option which are containing the ribosome. Okay. So ribosome is present in the option A. I will eliminate that. And ribosome is present in the option C. I will eliminate that. And in the option D, I will eliminate that. Okay. So which one is the left out option, guys? Only B, right? Yes. Option B is the correct answer. Okay, guys, give me thumbs up. Give me thumbs up, guys. If you understood this means. In the last class, I have told you, right? Is that centrosome is going to be present in the plant cell, guys? Can you tell me is that centrosome is going to be present in the plant cell? Yes or no? Is it, guys? Is it going to be present in the plant cell? No, very good. It is not going to be present in the plant cell. It is going to be present only in the animal cell. Remember, guys. Okay. So let us go to the eukaryote cell. Okay. So in the eukaryotic cell, the plant cells and the fungi cell which is having the cell wall, other than the plant and the fungi, the animal cell having only the plasma membrane. Okay. So each one organelle we are going to study now. First, we will overview about the eukaryotic cell. Okay. So we know that the eukaryotic is having the outer boundary which will separate the cell from the outer environment, right? So what is that we will call it as, guys? Plasma membrane, right? Plasma membrane will separate the internal environment of the cell from the outer environment, right? Yes. So inside we will come now. What are the things are present in the eukaryotes? All the membrane bound organelles, right? Except the ribosome, right, guys, and the centrosome. Yes. So you can see here the membrane bound organelle, we will go from the nucleus. Okay. So nucleus is there, right? So after the nucleus, the nuclear envelope continues with the endoplasmic reticulum, right? On the endoplasmic reticulum, if the ribosome is there, we will call it as rough endoplasmic reticulum because that is rough, right? Then coming to the endoplasmic reticulum, in some position, they are not at all having the ribosome. That we will call it as the smooth endoplasmic reticulum, right? So then we are having the Golgi apparatus, right, guys? Then we are having the lysosome, and then we are having the peroxisome, then mitochondria, right? And in the plant cell, we can see the chloroplast, right, guys? And other than that, important thing we have, that is the cytoskeleton, right? Yes, Nandesh, this speed is okay. Yes, guys, we are, yeah, because exam is nearer, we have to go in the fast only. So that's why I'm going in the fast. That is okay for you, right? Yes or no? Tell me in the chat box, guys. This is revision classes, right? You already studied this, right? Yes or no? Give me in the thumbs up or something, guys. Yes. So when we are calling about this all the eukaryotic cell, I have told you this, all the organelles are present. Now we will study each single organelle. Okay, fine. Yeah, let us come to the plasma membrane. Okay. So before going to talk about the plasma membrane, guys, I will ask you the random question to you, rapid question. The question is, who has discovered the dead cell first? Answer me fast in the chat box. Tell me fast. Who discovered the first dead cell, guys? Dead cell. YouTube student, answer me in the chat box. Robert Hooke. Very good, Robert Hooke, right? Yes. Then tell me who is the scientist who discovered the living cell? Living cell. Answer me fast. Answer me fast. Fast in the chat box, guys. Lakan, answer me in the chat box. Answer me fast. Anton van Leeuwenhoek, right? The scientist who discovered the living cell is the Anton van Leeuwenhoek. And another thing, the scientist who told that omnicellula is cellula, who is that scientist? The cell will exist from the pre-existing cell, right? Who is the scientist? Omnicellula is cellula. Who told that? Scientist name. Tell me, students. Rodolphe Virtue, right? Rodolphe Virtue. And the scientist who given the cell theory. Nandesh, answer me. Who is given the cell theory? Schilden and Chavan, right? Schilden and Chavan. Remember, guys, scientist names are important too. Okay, fine. Yes, yes. What do you mean by yes? Okay. So now let us go to the fluid mosaic model. Okay. So this fluid mosaic model will tell us about the structure of the plasma membrane. Okay. So this was given by the this was first given by the Singer and Nicholson in the year 1972. Okay. So in the year 1972. So they have told that. So in the plasma membranes are not arranged in the linear form, like, okay. So it is not like a uniform manner, okay, where lipids are arranged in the uniform manner. No, it is like the mosaic appearance. It is like a mosaic appearance. If you see this plasma membrane, it looks like the mosaic, right? The phospholipids are there, and in between the proteins are there, right? So it will look exactly like the icebergs in the sea, right? So how the sea was compared to the lipids in the sea of the lipid, where the proteins are looks like the icebergs, right, guys? Icebergs. So that is why the fluid mosaic model is compared to the see icebergs in the sea. Okay, fine. So first let us learn about the, and we will tell that this is not a solid structure. Okay, what we'll think, if the cell means it should be in the solid structure. No, guys, no. So the cell plasma membrane is not a solid, it is a quasi fluid. It is not a pura fluid also. It is like a quasi fluid, okay, or we will call it as a semi fluid in nature. Okay, then it is semi permeable. It's like you can see like a watchman near the gate. Okay, so he will not allow every person to go inside, right? So he will allow only the particular person to related person to go inside, right? So like that, the plasma membrane is semi permeable. Okay, fine. So then let us come to the composition of the plasma membrane. What are there in the plasma membrane? Okay, so what are there, guys? Answer me in the chat box. What are the things you can observe in the plasma membrane? Answer me in the chat box. What are the things you can observe in the plasma membrane, guys? Very good, Lakan. Very good. Rodolphe, it's you. Answer me fast. This is a revision class. Fast, guys. I need answer from everyone. This is a very important chapter. Sugar, yeah. Sugar, is that the main thing? Very good, very good. Yes, Nandish, protein. Yes. So what are the things, guys? We will see the phospholipid, guys, right? Then protein, right? Then we can see the cholesterol. Okay, let us see from the diagram. Okay. So here you can see it is having the phospholipid, right, guys? In the phospholipid, this end structure, we will call it as hydrophilic, right, guys? Hydrophilic means what? The head structure is the water loving. Means water loving means what? It will expose where the water is there. Okay, means in the cytoplasma side. Okay, in the extra cytoplasma side, the head is going to be exposed. Okay, so the head is consisting of the phosphate group. Okay, the head is consisting of phosphate group, right? Then coming to the tail. Okay, so the tail is hydrophobic, guys. Okay, so in the tail you can see one tail is saturated and another tail is unsaturated. Okay, so one tail is saturated and another tail is the unsaturated, right? So what do you mean by saturated? Can anyone tell me? Yeah, lover. Yes. So that is the water lover. Yes. Yes, what that? Yeah, singles bond. Yes, when we are talking about the saturated, here we can see the carbon-carbon, it is having the single bond, right? When we are talking about the unsaturated, it is having the carbon-carbon double bond, right? Yes, so it is having the double bond. Yes, so unsaturated fatty acid. In that also you can study in the lipids in the biomolecules where mono unsaturated fatty acid, poly unsaturated fatty acid, PUFA, MUFA, all the things will come. Okay, fine. So here you come to the tail. Tail is the hydrophobic. These are made up of the fatty acid, right guys? It is made up of the fatty acid. These are hydrophobic. These are not exposed to the water surface. These are hidden, right? These are the hidden inside. Here you can see these are hidden inside. Okay. So this is about the phospholipid structure. So then coming to the protein. So we will see the two types of the protein. One is integral protein. Okay. What is that protein, guys? Integral membrane protein. Okay. Here you can see these are integrated in the plasma membrane. Okay. These protein we will call it as the integral membrane protein. These may act as a carrier and these may act as a channel. You have studied in the transport in the plants, right? So these may act as a channel and this may act as the carrier. So these are helping the facilitated diffusion, right guys? Yes. So then there are the another proteins are there. So that we will call it as a peripheral protein. Okay. Either may it attach to the, if these are not embedded in the plasma membrane, it may attach to the extracellular surface or it may attach to the cytoplasmic surface. Okay. But these are not embedded in the plasma membrane. So that's why we'll call it as peripheral protein. They are in the peripheral. Okay, guys. So now we learned about the phospholipid and the protein, right? So now let us go to the cholesterol. Okay. So there are the cholesterols are there. They are in between the phospholipid tail. Okay. So where they are helpful in the stability of the plasma membrane. Remember, guys, the stability of the plasma membrane, stability of the plasma membrane is dependent upon the two things. Okay. So one is saturated or unsaturated. Okay. So one is based on the saturation. Okay. So if it is more saturated means, now you tell me, if the plasma membrane consisting of more saturated phospholipid. Now you tell me, saturated fatty acid means if it will become the solid or liquid. Tell me, guys. If it is contain the more fat, yes, Lakan, correct. If it is consisting of the more saturated fatty acid means whether it will become more solid or liquid. Tell me fast, guys. Tell me fast. It will become more solid, right? So you already know that coconut oil and the vanaspati ghee, these are the saturated fatty acid, right? So here also what will happen? So it should be not more saturated also and more unsaturated also. If it become the more unsaturated, it will become the oil-like structure, right, guys? So that's why both of the things should be there, but in the same, but in the different proportion. Okay. Both will be there, saturated and the unsaturated fatty acid. So based on the saturation and the unsaturation, the stability will be depend. And if the more saturated means stability will be decreases. If the more unsaturated means stability will be increases. Remember, guys. Okay. So then the second thing is the cholesterol. Okay. So the cholesterol was helps in the stability. Okay, because it will hold the tails. Okay, it will hold the tails of the fatty acid and it is helps in the stability. Okay, guys, remember that also. Okay, so then we will talk about the proteins which are attached to the, here you can see the proteins here attached to the carbohydrate. Okay, so that we will call it as a glycoprotein. The attachment here is the glucose, right? Carbohydrate, right? Carbohydrate, right? And here it was attached to the protein, right? So together we will call it as glycoprotein. Okay. So if you take the example where the glycoprotein is useful means you can tell the example of ABO blood groups. Okay. So they are having the different glycoproteins on the cell surface where they are helping in the identifying the blood group. Okay, guys. Okay. So here you can see if the protein was attached to the glucose or the carbohydrate, we will call it as the glycoprotein. Or if that here you can see for the lipid bilayer is there, right? For the lipid, if the glucose was attached or the carbohydrate was attached, what we will call it as glycolipid. Okay, guys, remember, if it is attached to the lipid, we will call it as a glycolipid. If it is attached to the protein, we will call it as glycoprotein. Remember, guys, okay? So then what are the things we have studied here? So in the phospholipid bilayer, we have the protein, we have the phospholipid, we have the glycoprotein, we have cholesterol, and we have the glycolipid. Okay, guys, do you understood this? And I have told you that yes, the transmembrane protein act as a carrier and the channels also. For example, if you take, not all the molecules will be permeable in the plasma membrane. So let us go to the permeability now. So which are the molecules can be permeable? Now you tell me, is that hydrophobic molecule is easily permeable through the plasma membrane? Tell me, guys. See, now I want to pass the hydrophobic molecules. Okay. So this hydrophobic molecule can easily pass through the plasma membrane, or it should be go through the integral protein. Tell me, guys. Tell me in the chat box. I need answer from you in the chat box. I will tell Lakan. Lakan, aquaporins is a channel, okay? So that is a protein which created that is the integral protein, okay? That is create a channel that is helping in the movement of the water molecule, aqua, right? Aquaporin means, so these will be channels which will help in the movement of the water in the plasma membrane, okay? Yeah, sorry guys, some network issue. Okay. Ma'am, you are there. Yeah, Lakan, some network issue ho gaya. No problem. So see here. So now you learned about the carrier proteins, right? So now the proteins, for example, some molecules which are the water soluble, okay? So which love the water, okay? So water molecules. Yeah, okay. Yeah, network issue. Fine, okay. So the molecules cannot pass through the plasma membrane, right? So which are the hydrophilic molecules, right? So for the transport, we have the integral proteins, okay, that may act as a carrier, that may act as a channel. So that will take the water soluble molecules from the outside to the inside of the cytosol, okay, inside of the cytoplasma. So that is the main work of the integral protein. Okay, guys. Remember always, you know that, right? So whenever they will ask in the hormone, in the chemical coordination, okay, the hormone is water soluble, okay? Can it pass through the plasma membrane? No, that require a receptor protein, right? That require the receptor protein. Receptor protein is nothing but the integral protein, okay? Fine. We can't chat in the Zoom. Is it Nandish? I will check. No problem, Nandish. You can answer here only in the YouTube. Okay. Fine. So now let us go to the protein. And I told you that in the beginning only the proteins are like the icebergs in the sea in the lipid. Okay. So and I have told you that the polar molecules cannot pass through the non-polar lipid bilayer. So they are the need the carrier proteins. Right, guys? Okay. So now the example. So here the example is the yeah, Lakan, I will send you 1 minute. Yes. Okay, 1 minute. I will send you. Lakanda, I have sent you. You can see. Okay, guys. So in that the example is the Na plus K plus pump. Okay. So remember Na plus K plus pump is the example for the carrier protein. Okay. So now here the structure is I have given. So the phospholipid head. Phospholipid head. Always remember, guys. It is consisting of the glycerol backbone, right? Glycerol is there, and for the glycerol molecule we have added the phosphate group. Okay. So if you study the biomolecules, in that the lipid they are given the glycerol structure. Okay. So in that phospholipid structure also they are given. You can see. Okay. So here you can see one tail is saturated and another tail is the unsaturated. Okay. So this is about the plasma membrane we have studied. Okay. In the plasma membrane, the lipid will show the two types of the movement. Okay, guys. That one movement we will call it as that is the flip-flop movement. Okay. So where, for example, if you see from here, the lipids are there. Okay, like this. So it will move like this. Okay, it will flip. Okay. This flipping we will call it as the flip-flop movement. Okay. And another is like this. One lipid is like here, and it will move laterally. Okay. So move laterally. So this movement is very frequent. Okay, lateral movement is very frequent. Then coming to the flip-flop movement. Okay, flip-flop movement is a very rare. Okay, so remember this, guys. The lipid in the plasma membrane will show the lateral movement and the flip-flop movement. Flip-flop movement is a rare. Okay, so but the lateral movement it will show frequently. Okay, fine. So student, do you understood this plasma membrane? Tell me in the chat box if you understood this. Padak, I have sent you. I have sent you here. I have sent you, Padak, the ID in the chat box. Okay. So now let us come to the cell wall. Okay. So you all know that where is the cell wall is present, guys. If you see the bacterial cell wall is made up of the peptidoglycan. In the last class we have studied, and also we will call it as a murin. Then coming to the fungi cell wall is made up of the chitin, right, guys? Chitin is a polysaccharide. Remember, guys, chitin is a polysaccharide, and we will call it as homopolysaccharide. Okay, we will call it as homopolysaccharide, and it is made up of the N-acetyl glucosamine. Remember, N-acetyl glucosamine. Okay, guys, it is made up of N-acetyl glucosamine. Then coming to the algae. So coming to the algae, so these are made up of 1 minute. Nandish, can you post the ID here? I'm posting ID, but it is not coming, I think. Okay, fine. So now let us come to the algae. Okay, in the algae, in some algae, like you can take the green algae, it is having blue-green algae that is having the cellulose, right? So it is having the cellulose and galactans and the mannose. Okay, so these are the cells. Cell wall is made up of bacterial cell wall, fungi cell wall, and the algae cell wall. Then coming to the plants, guys. Okay, so coming to the plant cell wall. So we are not going to in detail study of this of the cell wall. We are going to study of the plant cell wall here. Okay, in the plant, the cell wall is made up of cellulose, hemicellulose, and the pectin. Okay, guys, cellulose and the hemicellulose and the pectin. Remember here, when the plant was growing. Okay, so on that time, so in the early stages, it is having the primary cell wall. Okay, it is you can see the primary cell wall, like, so it is a green in color, right? So whenever it is in the green in color, you can see that cell wall that is having the primary cell wall. Okay, so later when it is grown, it is going to get the secondary cell wall. Okay, guys, when it is grown, then it is going to get the secondary cell wall. Okay, in the only in the early stages, it is having the primary cell wall. So let us see the structure of the cell wall. So here you can see this is the cell, right? Right, guys, this is a cell. And here you can see here I have written the benzene structure, right? So this is a cell, and near to the cell, guys, near to the cell, there is a secondary cell wall is there. Okay, so what we will think, ma'am, near the plasma membrane, the primary cell wall is there, no? No. So say primary cell wall is not there, secondary cell wall is there. Okay. So near here you can see the secondary cell wall, then you can see the primary cell wall. Remember guys, so this is the plasma membrane. Okay. So here you can see the plasma membrane here. Okay. And here you can see the nearly, here you can see the second one. So that is the secondary cell wall. Right guys. So then the last one. So last one you can see that is the primary cell wall. Okay, it is like this. Okay, so here you can see the plasma membrane, and here you can see the primary cell. Here you can see the secondary cell wall. Here you can see the primary cell wall. Okay, guys. So this is the plasma membrane. Okay, this is the secondary cell wall. Okay, secondary cell wall, and this is the primary cell wall. Okay, remember guys. Okay, fine. So here you can see the cell wall is the permeable. Okay, the cell wall is permeable in nature. Okay, fine. So in between the secondary, in between here you can see the middle lamellae. So the middle lamellae consisting of the calcium and the magnesium pectate. Okay, so calcium and the magnesium pectate, where middle lamellae is act as the cementing agent. Remember guys. Okay, calcium and the magnesium pectate, where it is act as the cementing agent between the cell or cell wall. And here you can see the secondary cell wall consisting of the suberin, lignin, and those are the non-elastic. So primary cell wall is the elastic guys. Okay, so they are not made up of the suberin and the lignin. Primary cell wall is made up of only the cellulose. Okay, so when you are talking about the secondary cell wall, it is made up of the lignin. Okay, so lignin and the suberin. Okay, so those are the non-elastic. So then coming to the endoplasmic reticulum. Okay, so you know that endoplasmic reticulum. Nandish, yeah, yes, Nandish. Password is 1234. After Lendo, this is the revision of the class. Okay, if you studied before, then only you can understand my idea, because we are going fast. This is a revision class. Okay. So let us talk about the endoplasmic reticulum. Okay. So guys, the endoplasmic reticulum, remember, it is the extension of the nuclear envelope. Okay. So here you can see the nuclear membrane is there, right? So the outer cell, outer membrane of the nucleus, which will form the endoplasmic reticulum. Okay. So here you can see outer cell wall of the nuclear membrane. Okay. Outer membrane of the nuclear envelope. So here you can see, so there are two types of the endoplasmic reticulum. Okay, so starting you can see in some endoplasmic reticulum there are the ribosomes are attached on it, right? So that endoplasmic reticulum we will call it as rough endoplasmic reticulum. Okay, because ribosomes are there, right? Then coming to the some of the endoplasmic reticulum does not have the ribosomes. Okay, that we will call it as the smooth endoplasmic reticulum. Right, guys? So now you tell me what is the function of rough endoplasmic reticulum and the smooth endoplasmic reticulum, guys? Can you tell me in the chat box? Lakan, meeting ID is shared, right? Okay. So when you're talking about the rough endoplasmic reticulum is involved in the synthesis of protein. Remember, guys, always. So this rough endoplasmic reticulum are involved in the synthesis of the membrane proteins. Now only we have studied about the plasma membrane, right? In that we have studied about the peripheral protein and the integral protein, right? So these proteins mainly synthesized by the rough endoplasmic reticulum. So rough endoplasmic, where the ribosomes are attached to the rough endoplasmic reticulum. Okay, so it should have to synthesize the protein, right? The ribosomes which are attached to the rough endoplasmic reticulum are involved in the membrane, involved in the synthesis of the protein which are membrane bound. Okay, fine. So now let us go to the smooth endoplasmic reticulum. Okay, we already told that smooth endoplasmic reticulum does not contain any type of the ribosome, right? So then what is the function of this? Okay, guys. So if you take the liver, okay, so in the liver, the detoxification process will done, right? We will take the so many drugs, right? We will take for example, if I Having the cold, I will take the tablet. If I am having the headache, I will take the tablet, right? So these are the drugs, right? How the detoxification is going to be done? The detoxification has to be done in the liver, right? So in the liver, in which organelle detoxification is going to be happen? That is none other than smooth endoplasmic reticulum, guys. Okay. So in the smooth endoplasmic reticulum, detoxification is going to be happen. Okay. The smooth endoplasmic reticulum which are present in the liver cell. Okay. Then what is the another function? The smooth endoplasmic reticulum which are present in the muscles. Okay. So you have studied about the sliding filament theory, right, guys? How many of you studied the sliding filament theory? Tell me in the chat box. How many of you studied the sliding filament theory, guys? Very good, Nandish. Yes. How many of you studied? Tell me in the chat box, guys. Raise your hands. Either raise your hands. How many of you studied? Yeah. How many of you studied about the sliding filament theory? In that, have you seen, guys? The calcium will come, right? The calcium will lead to the, it will remove the active. Yeah. Yes. Yes. So what it will do? It will remove the troponin, right? It will remove the troponin and the troponin on the active site of the actin, right? So it will remove the troponin. So then what will happen? So then the myosin head will come and bind, right? So the first what will happen? The calcium will come, calcium will remove, it will bind to the troponin, then troponin was removed, right? So then the myosin is going to be bind on the active side, right? So here it is helps in the calcium storage in the muscle. Okay, that's why in the muscle we will call it as the sarcoplasmic reticulum. Remember, guys. So, what is the name of the endoplasmic reticulum in the muscle means? So that is the sarcoplasmic reticulum. Remember. Okay, guys. So it is the storage of the calcium where the calciums are stored in it. Yes. So do you understood this, guys? And what is another function of the smooth endoplasmic reticulum? We have the phospholipid, right? So where the phospholipid is going to be synthesized, guys? From the smooth endoplasmic reticulum. Okay. So it is helps in the phospholipid synthesis, which is that smooth endoplasmic reticulum. Okay. Then another thing, it is helps in the synthesis of the some complex carbohydrate. Okay. So it is helps in the synthesis of complex carbohydrate. Complex carbohydrate. Okay, guys. Then it is also involved in the synthesis of steroidal hormones. Can you tell me which are the steroidal hormones? Can you tell me, guys, which are the steroidal hormones? In the chemical coordination, you have studied about that. Estrogen, progesterone. Right, guys? In the human physiology, you have studied about this, right? Which are the steroidal hormone? Progesterone and estrogen, right? Testosterone. Right. Yes, guys. So these are smooth endoplasmic reticulum is helpful in the synthesis of the steroidal hormone also. Okay, guys. Here you can see the structure. So here the endoplasmic reticulum is there, which is having the ribosome. Here is the endoplasmic reticulum is there, but does not have the ribosome. These are endoplasmic reticulum involved in the protein synthesis, and these are involved in the lipid synthesis. And one more thing, important function of the endoplasmic reticulum, I will tell you. Okay. So proteins are synthesized in the ribosome, right, guys? So see here. So in the nucleus, our DNA is there. So DNA is it was mRNA was synthesized, right? Then it was entered into the cytosol, right, guys? In the cytosol, what will happen? Protein synthesis will take place, right? Protein synthesis will take place. Then where it is protein synthesis is going to be taken place. So that is endoplasmic reticulum, that is attached to the where the ribosomes attached to the endoplasmic reticulum, right? So listen here, what will happen? The ribosome which is attached to the endoplasmic reticulum will synthesize the protein. Okay, so then it will send the proteins to the lumen. Okay, it will send the proteins to the lumen of the ER. Okay, so in the lumen of the ER, what are the changes will take place? Means in the protein we have the foldings, right, guys? So we are having the tertiary structure and the quaternary structure. The foldings are there, right? The here what will happen? So the foldings of the protein will happen in the endoplasmic reticulum. Okay, foldings of the protein will happen in the endoplasmic reticulum. Okay, the modifications. Okay, guys. Folding of protein. Okay, fine. Remember this, guys. Okay. And you have know the disulfide bond. So these all the things should be converted that primary structure of the protein into the secondary, tertiary, and the quaternary, right? So these all the things, okay? Formation of the disulfide bond and formation of the different bond will take place in the endoplasmic reticulum. Remember, guys, okay? In the endoplasmic reticulum, the formation of the bonds and everything will take place. Okay, not the peptide bond. So this is about the endoplasmic reticulum. Do you understood this endoplasmic reticulum? Give me thumbs up if you understood me. Give me thumbs up in the chat box, guys, if you understood means. Then we will go to the next. Fast, guys. Give me thumbs up. Now I will ask a question to you. Which one is the endoplasmic reticulum which is involved in the lipid synthesis? Can you tell me answer to this question? Which is the endoplasmic reticulum involved in the lipid synthesis? Answer me in the chat box, YouTube students. Fine. So now let us go to the Golgi apparatus. Very good, Nandish. Very good. Smooth endoplasmic reticulum. So now let us go to the, what is that? Our transport system here. Okay. So we will call it as a post office, guys. Okay. So here Golgi apparatus, we will call it as the post office. Let us study about the Golgi apparatus. Here the Golgi apparatus, like the tubular structure you can see, right? Tubular like structure. So each structure we will call it as the cisternae. Okay, cisternae. Okay, guys, we will call it as the cisternae. Okay, so this is here the what happened. The Golgi apparatus spaces. Here you can see the tubules, right? So these are some which is located near to the endoplasmic reticulum. Okay, for example, here you can see this is the endoplasmic reticulum. So some phase is nearer to the endoplasmic reticulum. Okay, this phase which is nearer to the endoplasmic reticulum, I will call it as cis phase. Okay, cis phase I will call it as. Okay, so or we will, it is the structure of the convex. Okay, guys, so we will call it as a convex, or we will call it as a cis phase. Okay, or we will call it as receiving phase, right? From the endoplasmic reticulum, it is receiving the protein, right, for the modification. So that's why this phase I will call it as a receiving phase. Okay, so now you can see, so these all are the compartments. Okay, these all are the compartments, and here you can see these all the things we will call it as a cisternae, right, guys? So here you can see the lumen. Okay, in the lumen of the Golgi apparatus, the modifications of the protein will take place. Okay, ma'am, all the modification has taken place in the endoplasmic reticulum only, right? So what modification is going to be taken place, ma'am? I will tell you guys. So remember, in the modification, what will happen here? The proteins will be added for the protein. The carbohydrate group will be added here. Okay. So now the proteins is converted into the glycoprotein. Remember, guys, always. Okay. The glycoprotein synthesis will take place in the Golgi apparatus. Carbohydrate attachment is going to be taken place in the Golgi apparatus, in the lumen of the Golgi apparatus. Remember this, guys. Okay. And the received, okay, the protein was received and it was modified in the lumen, right? So now it has to send outside, right? It has to be sent outside. So the protein will be sent outside by the maturing phase, guys. Okay. So here you can see the last phase here. Okay. So this last phase, we will call it as the trans phase or the maturing phase. Okay, trans phase or the maturing phase. From this, the protein will bud and come outside. Okay, it will come outside and it will reach to the particular position, or which is the particular location it will go. Okay, so that's why what we will tell. So our Golgi apparatus, it act as a post office. Okay, so just I will send the items. Okay, for example, if you take the Amazon. Okay, so Amazon, the producers are different, right? They will produce and they will send to the Amazon, right? So now what they will do? So they will send the, they will put the address of the client address, then they will send it to the people, right? So the same thing happen in the our post office. So that is the in the Golgi apparatus also. What will happen here? The protein was synthesizing and coming in the Golgi apparatus. It will put the address on it. Okay, it will put the address on it where it has to reach. Okay, so then it will send. Okay, guys, that is the one thing. And here you can see here acrosome formation will take place, and the plasma membrane making. Okay, and modification of the proteins and the lipids will take place in the where in the Golgi apparatus. Okay, so now what will happen? So yeah, so it will tag the particular address to the protein. So then it will send to the outside, guys. Remember, okay, fine. So this is about the Golgi apparatus. Okay, so do you have any confusion in the Golgi apparatus, guys? So here mainly you have to focus on the cis phase, that is the receiving phase, and the trans phase, that is the maturing phase. Okay, so what are, and it is made up of the cisternae. Some of them will confuse between the cristae and cisternae. Cristae you will see in the mitochondria. Cisternae you can see in the Golgi apparatus. Why we are calling as a Golgi means that is a scientist name, guys. Golgi. Okay, by the Golgi is a scientist name, so that's why he is given the Golgi. Okay, fine. And one more thing you have to remember, our lysosome. Lysosome is also made from the Golgi apparatus only, guys. Okay, it is made from the Golgi apparatus. Okay, fine. So do you understood this, guys? Give me thumbs up if you understood this means. Give me thumbs up, guys, if you understood means. Fine, fine, fine. Thank you so much. Let us go to the lysosome. Okay. So if you're talking about the lysosome, so where the name only telling that lysis, right, guys? Lysis. So lysosomes are single membrane. Okay. In the last class, I have told you which organelles are single membrane, which organelles are double membrane, right? Golgi apparatus is also single membrane, right? Okay. So lysosomes are the single membrane. Okay, guys. Lysosomes are the single membrane, and in the lysosome, it is consisting of all the hydrolytic enzyme. Okay, all the hydrolytic enzyme which function in the acidic pH. Okay, so which function in the acidic pH, the pH will be 4.5 to 5 pH. Okay, that is the acidic pH. And what is the main function of the lysosome? The lysosome is having the which is having the carbohydrate enzyme, which will break down the carbohydrates. Okay, and it is having the protease enzyme, which will break down the protein, and it is having the lipase enzyme, which will break the lipase, and it is having the nuclease enzyme. It will break the nucleic acid. Okay, fine. So this is about the thing. So you all know that the lysosome is called as a garbage, right? We will call it as what we will call it as, guys? Suicidal bag, not garbage. Suicidal bag, right, guys? Suicidal bag. Why we will call it as a suicidal bag? See about why we will call it as a suicidal bag. I will tell you. So, have you learned about the macro? Have you learned about the phagocytosis, guys? Phagocytosis in the human health and disease. Our macrophages are there, neutrophils are there, right? So, how the phagocytosis will happen, I will tell you. Okay. So now the neutrophil or the macrophage is there, so that will engulf the bacteria. Okay. So when it will engulf the bacteria, see here what will happen. Okay, it may be a food vacuole also. Okay, so see here what will happen when it is engulfs the bacteria. So it will fuse with the lysosome. Okay, so it will fuse with the lysosome. Then we will call it as a phagolysosome. Okay, so when it is engulfs the bacteria, then we will call it as a phagosome. Okay, so after the engulfing the bacteria, it will fuse with the lysosome. Then we will call it as a phagolysosome. Okay. So obviously, when the lysosome was fused with it, it is having the acidic pH and all the hydrolytic enzymes are there. So it will break down the proteins and the lipids which are present in the bacteria. Okay, guys, are you agree with this? So now what will happen? Through the exocytosis, What do you mean by exocytosis? Endocytosis, you all know that, right? Endocytosis means engulfing. Okay. So then exocytosis means releasing outside. All the breakdown product will be released outside from the macrophage. Okay. So this is how our lysosome, so that is a suicidal bag, is helping. Okay. Do you understood this, guys? Do you understood this? Tell me yes or no. Very good, Nandish. Yes, due to the digestive enzymes. Yes. So now let us go to the mitochondria. We have the two semi-autonomous organelles, right? Why we are calling as a semi-autonomous? Because the mitochondria is having its own DNA and own ribosome. Protein synthesis machinery it is having own, and it is having its own DNA, right, guys? But what will happen? All the proteins which are necessary for the mitochondria will not be synthesized in the mitochondria only. Okay. Some of the protein and some of the enzymes will be synthesized from the nuclear genome. Okay. From the nuclear genome. So that is the reason we will call it as semi-autonomous. Okay, guys. Fine. So now let us talk about the mitochondrial structure. It is having the double membrane structure. It is having the outer membrane and it is having the inner membrane. Right, guys? The outer membrane is, we know that outer membrane is the what permeable, whereas the inner membrane is the not permeable. Then coming to the inner membrane are having the inner membranes are having the proteins which are involved in the electron transport chain, right, guys? When we are talking about the electron transport chain, we have studied about that, right? Yes. And coming to the inner membrane, they are forming the folding like structure, inner membrane. Okay. So that we will call it as a matrix. Okay, guys. So for example, if you think this is the outer membrane, okay, this is the inner membrane. Inner membrane is forming the folding like structure, right, guys? This folding like structure, what we will call it as cristae. Sorry, what we will call it as? Yes, cristae. Right, we will call it as the cristae. Okay, and the inside the fluid. Okay, inside the fluid, we will call it as a matrix. We will call it as a cytoplasma, right? Like that. Inside this cristae, we will call it as the matrix. Okay, guys, matrix. So now the space between this inner membrane and the outer membrane, we will call it as intermembrane space. Okay, guys, intermembrane space. The inner membrane is having the proteins which are required for the ATP synthesis and the electron transport chain. Okay. So now let us come to the inner side. Okay, that is in the matrix. In the matrix, it is having DNA. Which DNA it is having, guys? Tell me. Whether it is having the linear DNA or whether it is having the circular DNA. Tell me, guys, whether it is having the linear DNA or it is having the circular DNA. It is having circular double-stranded DNA. Remember, guys, our nuclear genome consisting of the linear DNA. When we are talking about the mitochondria, it is consisting of the circular double-stranded DNA. Okay, circular double-stranded DNA. Circular double-stranded DNA. Okay, so then coming to I have told you that cristae, it will increase the surface area, and matrix is the site where the respiration will take place. Okay, guys. Remember, matrix is the site where the respiration will take place, and it is having all the enzymes of the electron transport chain except the succinyl dehydrogenase. That is complex number 2. Okay. So now let us come to the ribosome. It is having the ribosome, right? Which type ribosome it is having? That is having the ribosome, that is 70S ribosome. Okay, guys. And you know that. So it is having the ATP synthase. That ATP synthase is having the two things, two part. One is the F0 portion. That F0 portion is helping in the proton pumping, right? The F0 portion which is helping in the proton pumping, where F1 portion is helping in the ATP synthesis, right? ATP synthesis. Okay, guys. So what will happen in the electron transport chain? So we will use the reducing, right? We will use the NADPH and the FADH. We will remove the electron from that and we will produce the ATP. Right, guys? So that is about the mitochondria. Remember, guys. Okay. The mitochondria is having the double circular DNA and it is having the 70S type of the ribosome, where inner membrane is having the electron transport chain proteins. Okay, fine. So now let us come to the plastid. Okay, so when we are talking about the plastid, so there are three type of the plastids are there, guys. Okay, so one we will call it as the chloroplast. You all know that what is the function of the chloroplast. So those are involved in the what? Those are involved in the photosynthesis. Chloroplast, because they are having the chlorophyll molecule, right? They are involved in the photosynthesis, right, guys? Now let us talk about the chromoplast. What will happen? The tomato, right? The green, when the tomato is in the green color, during the ripening, what will happen? So the chloroplast which is present, they will convert it into the chromoplast. Okay, the chloroplast only converted into the chromoplast, where it will store they're having the pigments like the carotenoids and the xanthophyll. Okay, chromoplast is nothing but they are come from the chloroplast, converted from the chloroplast to the chromoplast. Okay, coloring agent, right? Yes. So these are having the carotenoids and the xanthophyll. Carotenoids and the xanthophyll, guys. Xanthophyll. Okay. So that is about the chromoplast. Now let us come to the leucoplast. Okay. So in the leucoplast, by the name only we can tell that these are the colorless. Colorless. Right, guys? These are the colorless. Then coming to the leucoplast, these are having the three types. Remember, guys, these are having the three types leucoplast. Okay, so one we will call it as the amyloplast. What do you mean by amyloplast? This amyloplast is store the starch in it. Okay, starch. Then coming to the elaioplast. Elaioplast was store the lipid in it. Okay, so it will store the lipid. Okay, guys. So then coming to the alluroplast, it will store the protein. Okay, so remember, guys, because the so many questions are asked from this part also. Like this, they will ask the questions like this. Okay. Yes, Lakan, correct. Yes, circular double-stranded DNA. So the question may be asked like this. Okay. The plastid, which is the leucoplastid, which is involved in the storage of the protein, is called as. Can you tell me, guys? The leucoplast, which is involved in the storage of protein, we will call it as what? Can anyone tell me, guys, in the chat box? Fast. I need answer from you. Answer me fast in the chat box, guys. Which store the protein? Protein. You have studied about the seed coat, right, guys? Allurone layer, right? So remember that is a protein. Okay, protein. Yes. So remember that allurone, that is the alluroplast. It will store the protein. Okay. Then coming to the elaioplast, that is store the lipid. Then amyloplast, you can easily remember, that is starch. That will store the starch, guys. Okay, fine. Then coming to the ribosome, I will quickly tell about the ribosome. Sorry, yeah, here I have to tell about the chloroplast structure. 1 minute. So let us come to the chloroplast structure. You all know that chloroplast is the double membrane, right, guys? It is a double membrane, right? So here you can see the cytoplasma. The cytoplasma inside the chloroplast, we will call it as the stroma, right, guys? Stroma, right, guys? So then we can see the thylakoids are present in the inside the chloroplast, right? So there you can see the coin-like structure, right? So here you can see if it is the one, okay? So only one is there. Remember, guys, only one is there. Then I will call it as a thylakoid, right? Thylakoid. Okay. Okay, 1 minute. Yeah, one is there means we will call it as the thylakoid. If it is having if it is having the bundle-like structure, okay? This thing I will call it as a grana. Grana. Remember, guys, okay? So where our photosystem 1 and photosystem 2 are present on this thylakoid. Okay, remember, guys. So and here you can see from the one grana to the another grana it is attaching. That we will call it as stroma lamellae. Remember, stroma lamellae, where one is connected to the another grana. That connecting structure is there, no? So that connecting structure we will call it as stroma lamellae. Stroma lamellae. Okay. So you know that our chloroplast, is also having the circular double-stranded DNA of its own, and it is having its own ribosome for the protein synthesis, right guys? The ribosome is 70S type. Okay, ribosome is 70S type. The cytoplasm inside the chloroplast, we will call it as stroma. Okay, you know that where the light reaction will take place in the thylakoid membrane, right? So where the dark reaction will take place, so that is in the stroma, right guys? Yes. So then coming to the here, there is another question they will ask. Photosystem 2. Photosystem, I will write like the 1 minute. PS, okay? It will be easy. PS2, okay? So you know that PS2, it is present on the oppressed, okay? We'll call it as the oppressed region of the thylakoid. Oppressed region of thylakoid. Okay, guys? So whereas photosystem 1, it is present on the non-oppressed region of thylakoid membrane, thylakoid. Okay, guys, remember, so many questions are asked from this statement also. Photosystem 2 present on the oppressed region, whereas the photosystem 1 presents on the non-oppressed region of the thylakoid membrane. Remember, guys. Okay, fine. So let us go to the ribosome. You all know that ribosome is the protein factory, right? So there are the non-membranous. They do not contain any type of the membrane. Only the ribosome is made up of protein and RNA. They are made up of only protein and RNA, right guys? So when you are talking about the ribosome, we will see in the prokaryotes, 70S type of the, very good Lakan, yes. In the prokaryote, we can see the 70S type of the ribosome. In the last class, I have told you, yes means nothing but sedimentation coefficient, right? And in the eukaryotes, you can see the 80S ribosome, right guys? The 70S ribosome was divided into the 50S and the 30S. Don't combine these two. It will become 80. It is not like that. Okay. It is about the sedimentation coefficient. We are given the 70S. Okay. So in the 50S subunit, we will call it as the largest subunit, guys. Okay, here you can see in the diagram, it is the largest subunit. It is made up of 5S rRNA, 5S rRNA, and it is made up of 23S rRNA. Remember this, guys. So 23S rRNA is having the ribozyme activity. During the translation process in the molecular biology, you have studied, right? Molecular basis of the inheritance, where the protein synthesis will take place when the bigger subunit of the ribosome will bind to the mRNA, right? That is nothing because it is consisting of the 23S rRNA that is having the ribozyme activity or peptidase activity, which will form the peptide bond between the amino acid. Okay, ribozyme activity, guys. Okay, so or we will call it as 1 minute, or we will call it as peptidase activity, peptidase enzyme. Okay, then coming to the 30S, 30S is having only the one RNA, that is the 16S rRNA, 16S rRNA. Guys, why I am telling you this specifically means they will give the rRNA. Okay, so they will give the 5S rRNA, 23S rRNA, 16S rRNA, and the 5.8S rRNA. They will tell that. So which one is belongs to the eukaryotic rRNA? Okay. So that is 5.S rRNA. So that is the reason you have to remember the RNA names, that is the 5S rRNA and the 23S rRNA. Like that you have to remember which is present in the prokaryotes and which is present in the eukaryote. Okay. Then coming to the 80S rRNA, in the 80S ribosomal subunit, it is divided into the 60S and 40S. Right guys? In the 60S, that is the largest subunit, right? In the 60S largest subunit, it is having the 3 rRNA. One is 5S rRNA and 5.8S rRNA, and it is having the 28S rRNA. Okay. So then coming to the 40S rRNA, it is having 18S rRNA. I will give you tricks here, guys. Okay. So what is the tricks mean? You can easily distinguish between the 80, that is a eukaryotic RNA and the prokaryotic RNA if it is containing the 8. Okay. If it is the 8 is coming, because here you can see 18 is there, 28 is there, and the 5.8 is there, right? If it is the 8 is coming, remember that that is a eukaryotic rRNA. Okay, guys. 23 and the 16S is there, means that is the prokaryotic rRNA. Okay, guys. So that's it for today, guys. So let us learn in the tomorrow's class. So what is the homework for you while coming to the tomorrow's class? So learn and come. Okay, guys. So some 2 more organelles are there that we will complete, and then we will go to the cell cycle. Okay. Yeah. Nandish, which is not in the test book? Subunit, yes, Nandish. It is not in the subunit. It is not given in the test book, but they may ask you, yeah, in the some of the exam, in the NEET exam only they have asked that. So, which is the rRNA which is present in the prokaryotes and which is present in the eukaryotes. Okay, so that is the reason it is important to remember. Okay. Okay, guys, read well, prepare well, all the best. Tomorrow you are having test, right? All the best, guys.